So welcome everyone. Thank you so much for joining us today. My name is Meria Terzi, and I'm a Women Who Code Fellow at the Women Who Code Emerging Tech track. Today's session is Artificial Intelligence in Drug Discovery Applications. And I'm very excited to welcome our amazing speaker today, Mojgan Asadi. And thank you all for joining us today. Mojgan has finished her undergraduate in chemistry in 2018, and she completed her PhD at the University of Southern California in 2023. Currently, Mojgan is a postdoctoral scholar at Stanford, where she investigates the intersection of AI with drug discovery, with premises to reshape the future of healthcare. I will now pass the session to you, Mojgan. Welcome to our event. Please share your screen and introduce yourself to everyone.
Thank you so much. Can everyone hear me well? Okay, very good. So thank you so much, Maria. It's so nice to again back to presentation for you guys, and hopefully make it become helpful for everyone who wants to continue working on chemistry, artificial intelligence in healthcare. I'm going to today discussing more about drug discovery in different perspectives, not only computer science, but also physics as well as chemistry, because it's my training is based on physics and chemistry, and I want to show the importance also of having fundamental science base for that type of questions and that type of research field. So as Maria said, I did finish my undergrad in chemistry at City College of New York in 2018. I finished in 2018. I finished in May 2018, and August 2018, I started my PhD at University of Southern California, and I completely joined a Nobel laureate group and completely did my PhD research on computer. And for postdoc now, I'm doing more hands-on experiments. So I'm trying to explore also possibility that doing a research experiment can bring to our understanding for inhibition, enzyme inhibition. So let's begin. I'm going to, let me share my screen fully. Okay, very good. So I'm going to talk about the drug discovery process in two different aspects. One, I selected two main enzymes that I work with. One of them is a family of tyrosine kinases inhibition. The other one is a COVID-IMPRO inhibition, which I will go over detail of each one of them and why I select these two systems. The other one, after I explain the systems, I'm going through the artificial intelligence and the current approaches for of artificial intelligence for the drug discovery we have, and hopefully get into the conclusion. So first, let's understand what is drug discovery and why is important. Drugs mainly are inhibitors. So after this, in my talk, whenever I mention inhibitor, so know that I'm talking about drug. Drugs are mainly inhibitors that they inhibit their target. Their target can be enzyme, which this is the purpose of my talk. The inhibitors go inside of the enzyme and inhibit or prevent the functionality of this enzyme. And this is very important for a variety of disease. So when this inhibitor goes to the enzyme, they can either temporarily or permanently inhibit the functionality of this enzyme. And this is very important because sometimes in certain disease, we wanted this enzyme not to function anymore. This protein, we don't want it to work the way it's working, especially when, for example, in the process of signal transduction. Now, the first system I picked is a tyrosine kinase. Tyrosine kinase, it's a very big family of enzymes. It includes many different enzymes that facilitate signal transduction. Let's say that we have on the figure that we have on the right, we have a receptor that leads any signal through these tyrosine kinases, this signal can come through the cell and make for example, our body to respond to these signals. So they play variety of role in cancer and also in signal transduction cascades. So in order to understand how these tyrosine kinases work, it's a very important question. How these tyrosine kinases work? Do we have how their inhibitor works in their body, in their function, and how we can design a better inhibitor for this type of tyrosine, for this type of enzymes that they are very important. But since it has a very big family, I don't want it to make you guys, go over, like it become too much if I talk about all the enzyme of this family. In my talk, I'm going to focus only two enzymes. One is Bruton tyrosine kinase, and the other one is a T-cell tyrosine kinase. They are homologous, and they are two enzymes that they are very important for cancer. These two enzymes, as we can see in the figure A, they are superimposed. So their structure is very similar. However, when the drug comes, which is the blue here molecule, it goes to the bonding site, which on figure B, I try to show the zoom. When the drug comes, we have a residues that they are amino acids that they are in the neighboring. They interact with this inhibitor and certain reaction take place. When certain reaction take place, this inhibitor can inhibit the functionality or the activity of these tyrosine kinases. Okay, but there is a problem here. This drug or inhibitor that is in blue, the molecules that is in blue, needs to be selective for this bonding site, for these enzymes. What happens if it's not selective? So when it's not selective, it means that it can inhibit any other enzyme that will look like this one that we have right now. So it's very important that we design a drug that is selective for the target we are interested. And what is our target? In this case, is tyrosine kinase. So remember, they will compete with other, for example, substrate that they want to bind here. So we have to design inhibitor that become very potent or selective for this bonding activity, for this bonding site. This is a key point here. Designing a selective inhibitor, number one. After we understood how this inhibitor come to the bonding site, now we have to understand how the other tyrosine kinases with the inhibitor we know will behave with respect to this bonding pocket. So this bonding pocket include different amino acids. These amino acids, they can be hydrophobic, they can have charge that we can call it hydrophilic. So they have different behavior that they can respond differently with this inhibitor that is going there and binds there. I collected all the inhibitors we have. It's a y-axis. And all the tyrosine kinases enzyme we have, which is on x-axis. I wanted to understand how different inhibitor with different enzyme of tyrosine kinase family have they have different selectivity? If they have different selectivity, why? So we collected all the inhibitor and all the enzymes for tyrosine kinase for this family. We find out certain residues, N represent residue amino acid asparagine, and E represent glutamic acid, and D represent aspartic acid. We find out certain residues that they are close to the inhibitor will give different selectivity. I'm trying to show how much is important to know, even if we want to use drug discovery, artificial intelligence for drug discovery, to have some basic understanding of chemistry, to understand, for example, the active site in more detailed form before getting to more modern application like drug discovery, like artificial intelligence. Okay, So first, we wanted to understand how residue amino acids that we have in the active site lead to the different selectivity of this drug. Interestingly, we find out when we have aspartic acid in this position. So see here, I showed with asparagine 484 and aspartic acid 445. The position here, this amino acid, it's very close to where this inhibitor has a chemical reactivity. So we show that when we have aspartic acid, which is a charged residues, instead of asparagine, which is not charged residues, we have a significant different selectivity. But in order to be sure about if having different amino acid nearby active site also lead to the different barrier, we have to do quantum mechanic, molecular mechanic type calculation. What does that mean? It means that we have to understand if my drug comes to the active site, to my protein. Let's say that this is drug, it comes to the enzyme. What would be the barrier that this drug make a covalent bond to this enzyme and stay there? Because some inhibitor will stay in the enzyme, some inhibitor will leave. But what would be the energy barrier? Because a smaller energy barrier means that this reaction happens faster. However, larger energy barrier means that, oh, this happens very rarely. So in order to understand the energy barrier, we have to use quantum mechanic, molecular mechanic. It's something that we use it very often in drug discovery calculations. When we are using quantum mechanic to capture when this inhibitor comes to bonding site, what would be the energy barrier? How fast or slow this reaction can happen? And this understanding using quantum mechanic, molecular mechanic, it helps us a lot, because if we know the energy barrier, let's say that for the known inhibitor, we find energy barrier, then we can design an inhibitor that can have a lower energy barrier. So this understanding is a very important, crucial for designing new drug. And the scheme on the left is the scheme that organic actually mechanism that happen to make this reaction to take place, right? We have our amino acid, asparagine, water comes, certain proton transfer happen, and then eventually, as we can see, this cysteine is a cysteine that is in our enzyme, make a covalently bond to our inhibitor. Then inhibitor stay in protein and inhibit the activity of this enzyme. This is the key. And this is the same thing when we have aspartic acid. So I tried to show when we have aspartic and asparagine, different charge residues, they lead to the different mechanism. So I applied the same understanding for COVID. We know coronavirus disease is affect all of us. Sometimes some of us lost our loved ones, unfortunately, during this COVID-19. This SARS-CoV-2 virus, it hits 77 million cases. Actually, I believe it's more, but its number of the cases is increasing and increasing. But one thing that lead to coronavirus, it's very important. Actually, one of the enzyme that is very important for COVID-19, it's protease or main proteases. And it's a big target for enzyme design, for inhibitor design. M stand for main and pro for protease. So it's like tyrosine kinase. Mpro also is enzyme. Inhibitor go bind to the bonding site of Mpro and inhibit the activity of COVID. But the question is that, see, sometimes when inhibitor goes to the active site, either can leave or should stay. For COVID-19, we want to design an inhibitor that stay in the bonding site temporarily. because we don't want this enzyme to be available for other inhibitor. So we want to make sure this enzyme can get blocked and won't continue its activity. So for SARS-CoV inhibitor, we have two inhibitors that they are on the left. The red one is the place that I try to show it with red, is the place that exactly when it goes to the COVID, it binds to the COVID. The red one is, we call it as a reactive part. It goes to the enzyme. One we have hydrogen and the other one we have nitrogen. It leads to very different reactivity of inhibition. On panel B, we can see when we have hydrogen, how this, for example, MPRO has cysteine, will have covalently bond that can lead to the different exothermicity. Actually, our study is a collaboration with the experimental group that we found out. This is my PhD work, which is we are publishing it soon. We find out when we have hydrogen instead of nitrogen, we'll have different exothermicity. So the exothermicity of this one, for example, when we have hydrogen, it become not exothermic. Like it will, when we want to go return, it cannot return, stay there temporarily and cannot go back anymore. And we find that this is good understanding that we can design new inhibitors that have similar reactive group that stay in the enzyme forever and they won't leave. I mean, when I'm saying they stay in the enzyme, it still has a period of time. Still they can leave until after a certain amount of the time. So it's very important we know at what time scale the inhibitors they can leave. So this is the reaction mechanism that we have our different inhibitor, that one has the nitrogen, the other has hydrogen, and lead to the different mechanism. You can see that when we have nitrogen, water participate role. When we don't have nitrogen, when we have hydrogen, water does not participate role. And someone might ask why presence of water is very important. It does very important for these energy barriers, because when we have hydrogen, this energy, it become minus 35. It means that if this inhibitor binds to the active site, it makes it so difficult. It should go barrier of minus 35 to go back, to again remove itself from the bonding site. However, when we have nitrogen on the active site, it's only minus 1.6. It's very easy to go back again. So they make it as a reversible and irreversible energy calculation. So it is very important. Now, let's say that now we understand, okay, when we have hydrogen, it leads us to irreversible mechanism. Now let's design a new inhibitor that has the same property, but has better selectivity, that it become again irreversible to the bonding site of the enzyme. So with using quantum mechanics, we can have very solid fundamental thermodynamic understanding of the reaction mechanism, number one, how certain inhibitor with certain reactive group, when they come to the bonding site, behave, what is the reaction. Now that we have this understanding of known inhibitor, because these inhibitors that I show so far are known inhibitors. Now that we understand known inhibitor, the property, the energy barrier, thermodynamic, everything of the known inhibitor, now let's use this understanding to bring it to design a new inhibitor. So before getting to this part, now I wanted to see what we learned. We learned about the selectivity. So I'm going to over this a lot. So selectivity is a key here. Selectivity means that I design inhibitor for my enzyme that can only inhibit my target enzyme. It won't, because in our body, we have a lot of proteins, different enzymes, that we don't want to design a drug or inhibitor that also inhibits other enzymes that they are not our interest. On the other hand, it means that we want to drug enzymes inhibitor that has a less side effect. So this is the key. And I tried to show the use of the quantum mechanic for two different examples. One of them are tyrosine kinase, the other one is COVID. I showed the theory and computational behind the reaction mechanism and why they are important for understanding, for designing new drug. And now I'm going through the more modern research on drug discovery, which is artificial intelligence. So artificial intelligence, let's start off with this. Artificial intelligence, like many other things, is just tool. The question is important. So AI make it possible to learn from experience. So AI in any different field, if we use, it's used from what we experience. It use our data. So it use the data to perform a human task, because sometimes we have a lot of data, but as a human, if we go over each one of the data, it will take so long. So we can use a voice algorithm that can screen over our data and give us what we are looking for. One of the key that important factor that AI progress actually very well in drug discovery, one of them is using the screening, which we go in detail in next slide. And also the other thing is that optimal in the situation that we have, it's the problem with AI, unfortunately, is that works very well when we have a lot of data, because in some of our cases, we do have this problem that for certain drug, for certain body in our body, for certain enzymes and proteins that still experimentally was not invested well yet, we cannot also do anything with AI because we don't have big data. So this is one of the big problems. So for using artificial intelligence, first we have to design our molecular representation. So the drug we are looking for either can be present as, let me just put my highlighter, very good. So either can be represented as a graph, or can be fingerprints, can be a string, or can be 3D. It really depends what you are interested and what you're doing. So it's not like this one is better than the other. It's really depends on the question you're asking. Graph can be represented as a fingerprint, or also can be represented as the string. This is better for memory. Or also can represent, for example, 3D can be represented as a string, that in each one of them, when we are using artificial intelligence, we have V, which represent our vertices. It shows that the elements, so for example, let's say that we have oxygen and hydrogen here. E edges represent the bond between this oxygen and hydrogen, and V, which is our vertices, represent each atom. Oxygen, we have oxygen, and we have, for example, hydrogen. So number one, we have to show how we want to represent our molecule for our code. The other one is that we have a progress, actually. So in early, when we were using generative models, small format, which we use very small string that I wrote it here, smallest representation, we use it in the earliest stage, that it was the network similar to language model. But we have a very good progress. So graph neural network is which use a lot in enzyme in inhibitor design, drug design, and message passing network. Now they are more powerful approaches. The reasoning is that when we were using a small format in molecule, we always have ring, and when we were using small format, not necessarily we could get good information about the ring, cannot distinguish the ring. So now the approaches we have, for example, like junction tree of a variation of autoencoder, they have respect it's a better representative of the rings. So that's why I wrote it as a more powerful approach. So it's very important here that which approaches we are going to use for our molecular representation. So the other thing that AI it become very good at it is that, let's say that me as a user, I wanted to design a drug. I wanted to, example that I gave, tyrosine kinase or COVID. And I want to say that I found for the known drug that they have, for example, for tyrosine kinase, one of the FDA-approved drug is ibrutinib. Let's say that for ibrutinib, I calculate computationally and experimentally, and I didn't find a very good selectivity for this specific drug for the bonding pocket of my enzyme. Now I want to use artificial intelligence, and I want to say that, oh, I want to see from the data we have, which is a big library, one of them is zinc, the other one is QM9, which one would give me a drug which is better than what we have right now. So two library that I suggest here, as I said, is a zinc and QM, which has a million number of the drugs already, drug-like compound, but I will tell you what is the limitation with these two. But we can use for drug-like compounds that we can use artificial intelligence actually to screen through this million number of the drugs we have and to dock it through our target enzyme to find which one fits better in the pocket of the enzyme. See, if we do this one as a human task, it would take forever. So it's helpful now to use this library that we made it with the research. And now we search through which one would bind even better to the bonding pocket. So one of the method you guys can use is a graph convolutional neural network, which works directly structure, graph-like structure like molecules, and try to dock it. Actually works for protein ligand complex. It's very nice, and it's going to predict the bonding affinity. Because bonding affinity, I mean, is when you have a specific drug and you want to bring it to the bonding pocket, bonding affinity of this bonding pocket to this inhibitor is important to know how much, in the very simple explanation, how much they like each other. So it's very important when this one comes here, this enzyme like this inhibitor so much that won't let it to go. So this using graph convolutional neural network to predict the bonding affinity, it is very important. So one that I said I want to go over the limitation. One limitation with zinc and QO9 are the following. Like many other tools, AI has its own limitation. As I mentioned earlier, one of the limitation is that we don't is the data. Okay, so let me go detailed explanation of one zinc and QM9 limitation. When we are dealing with drug, there are two type drugs. Drugs that can come without any chemical interaction, like bonding, a specific expensive bonding to the inhibitor, into the enzyme, can just come here, stay here, and then leave. We call this one drug-like compound. But sometimes when the inhibitor comes, it bonds covalently, very strong bond to here. Unfortunately, zinc and QM9, they don't have good data when this inhibitor comes and bond. They have a good data when it comes and just interact electrostatically van der Waals here. So that's one of the limitations that I'm sure with the progress that has in this type of calculation, I'm sure it will be, this data also will be available soon. But yeah, one of the limitations, we can only predict the drugs that they are not covalently interacted, so that they can come and easily leave. So the other, so let's say that now you went through a graph convolutional neural network, you found the drugs you want. But then you need to go over a screening method. We call it as a post-screening actually method, which is finding the bonding affinity, right? There is two ways. If you have 2 or 3 drugs, so simple. You can use Schrödinger, you can use autodock to dock your inhibitor inside the this enzyme. So simple. It's not very expensive. It's just 1 or 2. It takes 20 minutes each. So 40 minutes calculation for 2 inhibitors. But let's say that from this data, you found 100 inhibitor that you want to see the bonding affinity. You cannot do it one by one, right? So there are good method, deep learning method, EquipBind, for example, that again use neural network that is going to predict for us the bonding affinity and predict for us actually the good bonding pose. Because see, when the inhibitor comes to the active site, how this inhibitor rotate, what is the pose also is very important. Because there are in the active site of the enzyme, there are many amino acids that with the pose can give different energy. So this pose also of inhibitor is very important. That's why selecting wisely the docking algorithm also is important. So with all that being said, yeah, I want to use, I was hoping actually in my talk to show artificial intelligence in the drug discovery, not only as a computer task that blindly can be run on any data. I was hoping to get in more detail also physics and chemistry background first before getting to the artificial intelligence part, because to my perspective, this one is way more important. To know the question you are asking, how important it is, because it's related to human health. So we should be more critical the way we do research when it comes to human health. I mean, in everything, but when it comes to human health, I think we have to be very careful. So the goal is that we try to use quantum mechanics and the physics behind the quantum mechanic and theory to find out what is the correct, what is a good selective, not toxic inhibitor for our target enzyme. So even though we have a lot of actually improvement using artificial intelligence and even quantum mechanics in designing new enzymes, we have still need improvement. As I said, for example, we still need improvement when the drugs bind covalently to the enzyme. So I hope the people who wanted to start in this field, they get a fundamental understanding of math, chemistry, and computer science, and then computer science. And yeah, so hopefully in future we have people who are working more on inhibitor design, that we can design drugs that they have less side effect for the patients. So with all being said, I want to thank you again, everyone. And anytime you guys ask, you have question for anything, for code, for chemistry, math, physics, feel free to email me. And yeah. And
Mojgan, thank you so much. Your presentation was super interesting. And thank you for providing this. We do have one question, a starting question. I have also a few questions I would like to ask with you. So the first one, okay, someone has no background on chemistry or biology. Maybe she has some coding experience and she's interested about this domain. Are there any resources available that she can start with? Maybe a book or an online course or some seminars she can watch? What do you think?
So that's a good question. It really depends where she wants to start. If she wants to start from understanding more quantum mechanics, because my advisor in PhD his Nobel Prize is because of the quantum mechanic, molecular mechanic type calculation. I can introduce you his book, which I really, not because he was my PhD advisor, but it's a very, has a very fundamental physics for understanding quantum mechanic. So it really, this person, she wanted to start from the physics part of calculation. For example, if someone is interested on quantum mechanic, yeah, definitely I can introduce the book, some books, which I think anyone who wants to enter this field, I suggested to start from quantum mechanic part, Gaussian, DFT type calculation. Know the level of theory, know how to reproduce experimental calculation. Especially if the person has machine learning background, that doesn't need to spend time on learning how to code. I really think starting from understanding the mechanism with using quantum mechanics, I think is a starting point.
So I was wondering if there are available data sets that you need to train models to predict things. Can someone build their own model, like machine learning model, and try to get a better accuracy on the predictions? Is there any research on that? I mean, that we can or are there any Kaggle competitions related? I don't know if you're familiar with anything that you can share.
Yeah, that's, recently we are working on one system, which is all those reductase. I don't want to, because it's recording, because this work is not, it's very, yeah.
We can talk about other research or relevant.
Yeah. So it is possible. So see, the data that we have, sometimes when we do screening, it happens that they give us inhibitor that they are so off, we are not even looking for. It's something that when you look at it, like the structure, it's so bulky sometimes that does not even be able to fit, for example, for the bonding pocket. And sometimes let's say that we end up with the good drugs, but you're saying that, no, still I'm not getting, you run it through the affinity, bonding calculation affinity, and you find out none of them give you a better, good affinity. You're not happy what you got from the data. It is possible to design your own drug, but the problem is that you have to run it through synthesizing it. Because if you want to send any drug for FDA proof, you have to prove that this drug, you synthesize it, and it can work well in the experimental lab, in the wet lab, with your enzyme. It becomes very expensive calculation. It means that because it's a try and error, you have to try it, you have to see if it works. Again, you have to back to the lab, you have to work again and try it. So, but I know the people who manually design inhibitor and they send it for synthesizing. But yes, it is possible.
Great. Someone has asked for the titles of the books. If you can share them either in the Slack in the chat here, or if it's easier for you, you can put it in the Slack of the emerging tech track. If it's easier for you to find them later, I mean, if you don't have them, you can't. And my next question for you, can you describe like a typical day in your office? So you do research on drug discovery using AI.
Right.
What are you working with? Are you writing code? Are you researching papers? Is it MATLAB? How does it work?
So I did. I woke up early morning. So let me say from beginning when I woke up. So I woke up very early morning, very early. So that's why 7 a.m. for me for presentation was not a big deal. But I woke up early, and then after I do some task, I go to my office. The office is a wet lab, but I'm doing both computational and experimental work. And the reason is that I didn't want to only enhance the skill of my computer science. I wanted to, because my PhD was dual degree, was PhD in chemistry and computer science master. So I didn't want to only stay in computer science and sit behind the computer. So I wanted to be in the lab to do also experiment. Because so many things when you do code, you don't understand until you go especially when it comes to inhibition assay. So now when I go, I do some experiment and I spend again a few hours on computer. But the lab right now I'm working is a vet lab. And the people do experiment work. And we work with both soluble proteins, the protein that they are surrounded with water and with solvent, or the proteins that are surrounded with lipid. And in some cases we also would work with viruses. But yeah, now I'm in the vet lab, and then until like late night. I don't stay in the office until late night. I stay in there until like 6-7 p.m. and then I go home, play some piano, and then.
That's great. I can see that there's a lot of interest on the machine learning side. So
I'm sure.
Girls here know coding and they're interested in machine learning. So I would like to ask, in your research group or at the university, is there any collaboration between pure computer scientists focused on machine learning and people who understand about chemistry and biology? Is there any case that they do research together? So one brings the machine learning expertise and the other one brings the chemistry and biology expertise, for example.
Yeah, right. We have Stanford actually is very collaborative environment, has a very collaborative environment. We don't know everything. So it's always very nice to learn from different people. And that's the whole point of collaboration. I'm always so open about the collaboration. But yeah, we do have very strong collaborative with the computational people, we have with the experimental people, we have with computer science department. So yeah, physics department actually one of the projects in the lab. So there are, we do have close collaborators with different disciplines, because it does help also to grow. But the question is if we can find a common language, because you see the people who work on computer science, not necessarily they might talk about the same thing, but they not have the same language as a chemist who talk about the same thing. So it's just your task to be clever enough to find a common language that both can become a win-win project. That's the only difficulty. But beside that, we are always open for collaborating and nice. But also I found the paper, the name of the book. So I share it here.
Great. That's amazing. Thank you so much for providing this. And I have another question. So you said that you trained a neural network, like a deep learning model, or people are using deep learning models to discover new drugs and see how effective they are. In which environment are they building these models? Are they doing like a Python script? Are they using any Python libraries? Is it on another tool that we are not aware of?
No, it's on Python. So the whatever happened recently is in Python. I mean, except Julia, but it's Python. But when I talk about QM, we do have also Fortran. Sometimes the implementation are over Fortran. But for machine learning, artificial intelligence, whatever I use so far for my research, not only for drug discovery, even for different tasks, for enzyme design also, they are in Python. So it make it easier actually for new generation to be able to implement.
Do you have any libraries that you use often?
Library for
Yes, like Python libraries, for example, sklearn or TensorFlow, or
I mean,
of
course,
in other tasks we use TensorFlow.
TensorFlow is something that TensorFlow is what I'm PyTorch sometimes, but we use mainly TensorFlow. But but you know what? Something that I want to say is that if someone knows, I never learned Fortran, for example. Our generation, I don't think so. But I think if you know one language well, it won't become difficult to because in one of my projects for my PhD, I needed to implement something in Fortran for QM. So I never in my life learn or even code Fortran. But if you know one language well, it's faster. I'm not saying it's easier. It's faster to also implement or read and debug other languages. So don't jump around. So it's fine to know only one language so well, one library so well, one like even Mathematica for plotting. Sometimes we use the people jump a lot from MATLAB, Mathematica. So it's fine to know one thing well, and use what you're comfortable with.
Great. Thank you. Thank you so much for your advice. I believe it's going to be extremely valuable for anyone that is starting now in machine learning or coding and wants to have use drug discovery as an application domain as well. Why not? As a final question before we close this session, I would like to ask you, what do you think about the future? Do you think that the new generative AI or the big large language models that we have now are now being used in other domains are going to be used for drug discovery? Do you think that there will be a day that we will discover drugs for everything faster than ever before? Or there are still challenges that
no, there are. I think we have a long way to go there, in my perspective, because still I think there are many challenges that we have for using AI for everything. First, let's say that especially when it comes certain type of enzyme in our body that we don't experimentally, we don't have good understanding. I don't think so AI can have a good understanding. So still I'm hoping that more and more people are coming to this field. They work, especially females, they work on these tasks. So then we can generate libraries for AI to give to protect drugs for the other drugs enzymes that we haven't worked on. But for example, especially when it comes to enzymes that they are embedded in lipid, the understanding, like alpha-4, for example, we do predict well the enzymes that they are in the solvent. But when it comes still to this enzymes that are embedded to the lipid, still we have a long way to go. So imagine we are not in the stage to predict the structure, forget the product, the inhibitor. So and it's like a building, it's like making a building. So you have to do one by like step by a step. You cannot jump to the roof. So but in future, things going too fast, but I'm hoping more people with more fundamental knowledge in turn come to this field. Yeah, and they might do amazing things.
I couldn't agree more. And I have one more question. So you said at the beginning of your talk and during your talk as well, when talking about machine learning and AI and how it can be used for drug discovery, that one of the biggest challenges is data.
Mm-hmm.
How can we solve this? Do you think that they can put like sensors in our body, or somehow medical devices, we will be using more and more medical devices in order to track and better understand the application of new data, of new drugs?
Okay, so the data problem, it comes from experimental lab. Especially now I join experimental lab. I appreciate more the expensive the experiment they're doing. Expensive, I'm talking about the human time. So I think the problem comes from the experimental lab because so many things in the lab, in experimental lab, still are done by human by doing experiment. So it's very time consuming, number one. Number two, there are errors can happen because human sometimes is tired, sometimes they are not focused. In the lab, many mistakes can happen. When mistake can happen, it means that you have to go back again at the beginning and do the experiment again. So I'm hoping that we will have more artificial intelligence designed for human task job in the wet lab first. That make the progress faster. Then we will have more data. So
interesting.
yeah, because I see, for example, as simple as protein purification, that when we want to solve a protein, structure of protein, you have to purify protein to get rid of everything that your protein was growing to have your pure perfect protein. So human does that. And I saw it that they do it a lot of mistake. They don't know which column to pick. They don't know what the buffer they have to use. So I was thinking by myself, oh wow, if we can use also the data they already have for all these wet lab, and we can also design an algorithm for these people instead of doing it, finding mistake, go back again at the beginning, do a whole experiment. And at the end, what is leads to less data for us. So I think that we can design robot or something. I hope none of my experimental friends listen to hear this, but we can design robot that they can do that type of experiment. Then it become faster because they do less mistake. It become faster, and then they generate more data for us. Then this data we can use this data for hopefully designing new things that we don't know, like new drugs.
AI opens so many new ways, and I do personally think that it's gonna be catalytic in the evolution of the drug discovery domain. And I would like to thank you for your session today. I think you have opened our eyes a bit more on this field, and gave us a good understanding of how drug discovery is taking place. And also, I think you inspired me personally, and I think the audience as well, on how we can use AI either in discovering new drugs or predicting the efficiency of drugs, and even finding new ways that we can work in this field or build things in this field, either for collecting data or validating data or training models that can be beneficial. So I would like to thank you again on behalf of everyone. Your final remarks or something final you would like to share with the audience again today?
So the only thing I want to say for the females that we have here, try to learn math as much as you can. Trust me, try to learn math, because even if you want to be expert in machine learning, if you want to be expert in artificial intelligence, if you want to be expert in any of the thing I explained right now, you have to train your brain in the math. So just learn math as much as you can. Read as many books as you have. There are so many free sources online. So just stay tuned with math. Yeah.
Thank you so much, Mojgan. Thank you everyone who joined today. Please, you can follow us on Slack. Please check out the next events that we have on AI. This track, the Emerging Tech track, is gonna put a focus on AI for the next coming months. And enjoy your day, your evening, your afternoon, wherever you are. Thank you, everyone.
Yeah, thank you. Bye-bye.